Hi, I'm Jasmine McBelly, and today we're going to talk about cell organelles. Did you know that here on the International Space Station, we investigate the inner workings of thousands of cells? Let's go. The different parts of cells can be compared to the many parts of the space station. But before we do that, we need to know what cells are. Cells are the basic building blocks for life as we know it. Living organisms can be made up of a single cell or millions of cells working together. Cells of complex organisms have inner parts called organelles. Just like each element of the space station has a specific function to keep operations running smoothly up here, organelles have specific jobs to do for their cell. The tranquility node could be considered our nucleus here on the space station because it contains the life support and environmental controls needed to make the ISS habitable. Likewise, the nucleus of a cell contains the genetic material and control center for a cell's processes and functions. The space station is surrounded by an exterior barrier that keeps the inside of the space station safe from the external hazards, like extreme temperatures, UV rays, and micrometeorites. Docking stations allow vehicles to visit the ISS with a resupply of essentials, like food and supplies sent from Earth. Similarly, the cell membrane has channels and pores built from protein molecules to allow certain substances to enter or leave the cell. The solar arrays on the space station can be adjusted by our mission control team to maximize the amount of sunlight they receive. Like the chloroplasts of plant cells, the solar arrays convert sunlight into usable forms of energy. When the space station is in sunlight, the electricity generated by the arrays is used to charge the station's batteries. The batteries power the station, just like the mitochondria powers the cell. The atmosphere in the space station can be compared to the cell's cytoplasm, which is the fluid that fills the cell, contains the organelles and cytoskeleton, and allows intracellular movement of organelles. The atmosphere on the space station allows for the movement of astronauts, supplies, and equipment on station. Trusses on the space station provide support and stability for the various modules, much like the cell's cytoskeleton provides support within the cell and is made of interlinking protein filaments. The zariye module, or functional cargo block, was the first module of the space station to be launched, but it's mostly used for storage now. Similarly, cells use specialized compartments called vacuoles that are storage spaces for cellular material, waste, and water. Here on station, we use cargo transfer bags, also known as CTBs, to conveniently store equipment, hardware, and tools. CTBs allow all these loose items to be transported from module to module efficiently, as needed. Similarly, the Golgi apparatus processes and packages proteins in a cell. The recycling system on the space station breaks down waste and enables reuse of critical resources, including water and oxygen. These resources can be difficult to transport to station. Likewise, lysosomes in a cell break down and recycle waste and pathogens for use in the making of new cellular parts. The space station is a unique laboratory that makes it possible to study cells without constraints of gravity. The BioServe microscope was specifically modified for use on the ISS. It allows astronauts to capture digital, high-definition microscopy images and videos of experiments. These images are then sent back to Earth for analysis by scientists. Thanks for learning about the parts of a cell and the space station with me today. The results from research on cells conducted here on the ISS will help us better understand the human body and solve biomedical problems that occur both on Earth and in space. Think about the many roles your cells play to keep you alive and well. Learn more about cells in the classroom connection that accompanies this video at nasa.gov/STEMonstrations. See you next time.